1D Research Consortium together, and we've got more people coming in all the time, which is great. So I'm going to try sharing my screen and just checking. Oh, wrong way round. Is that the wrong way round at the minute?
Yeah.
Yeah. Okay. Let's do.
It's the little dots in the circle. That, I think that means you can change
to
it.
display settings.
Oh yeah.
Yeah. And then click it there.
Yeah. There we go.
Yeah.
Usually I'm fine dealing with that, but for a minute it just couldn't see it. So I'm going to pop that up in the top corner there. So can you see that all okay now?
Yeah, it's lovely. Thanks.
Okay, fine. And I can't see you just because of the setup of the thing. So if anyone needs to stop me, just raise your hand or wave at me or shout. I'm happy to be interrupted. So I thought what I'd try and do today is give you a quick overview of the Type 1 Diabetes Grand Challenge that's been funded by Steve Morgan Foundation, JDRF and DUK. So a little bit of information about that. And then I'm going to tell you a little bit briefly about what my fellowship is focused on. And I'm going to sort of finish with telling you some of the really interesting results that are starting to emerge and what we think the impacts of those might be. So see if we can get this. So I can move things. So I'm going to be telling you the title of my fellowship initially is the Leveraging New Knowledge from the Human Pancreas to Advance and Improve the Understanding and Treatment of Type 1 Diabetes. And you all know where I work and you can get hold of me if you want to ask any further questions. So just to kind of put this all into context, this is Steve and Sally Morgan and Sally's son Hugo, who developed type 1 diabetes at the age of 8. And like everybody who's diagnosed or has a family member who's diagnosed with type 1 diabetes, their life changed from that day forward. But Steve and Sally have been incredibly generous and have given £50 million, which is a huge amount of money, to type 1 diabetes research in the UK. And they have helped with JDRF and Diabetes UK to set up this type 1 diabetes Grand Challenge. And there are 3 main areas in which they have invested that money, which you can see is sort of highlighted down here. Can you guys see my let me see if I can put my pointer on so you can see that. Can you see the pointer now?
Yes, we can.
Yeah, perfect. So there are 3 main kind of areas in which they're focusing the funds. Root causes, looking at generating new and improved beta cells for treatment, and novel insulins, so trying to generate better insulins for treatment. If you want more details about the Grand Challenge, that's included here. And essentially, the initial part of the funding went out for 3 Steve Morgan Foundation Grand Challenge Fellows, Senior Research Fellows, and I'm one of those, along with Vicky Salem and James Cantley. And then since then, they have put out several calls for particularly kind of in root causes and in immunotherapies, a big call looking at how we can generate stem cell derived beta cells and improving the way we can do that, and a further large call on novel insulins. And there's more recently been some funding out there for innovative sort of high risk grants that I know several people from within the consortium have been successful in getting. So this has been a huge investment of money in the UK for type 1 diabetes researchers. And on a slide I'll show you at the end, I really think it has brought the community together, which has been fantastic. And I really hope to see that grow over the next little while. But this is something that kind of Steve mentioned at the beginning, and both Steve and Sally, that they were really insistent that from day one we needed to work together. They were investing this money to get us working together, no more working in our silos, trying to work as a team. The enemy, of course, out there is type 1 diabetes, and by joining ourselves together and attacking as a team, we've got a better chance. So we've been really pushing this forward for the last year or so now. More people are coming on board. We've got a a consortium meeting in November, which I'm really looking forward to, where we're getting everybody together to discuss some of the research that's coming out. So it's a really exciting time, I think, to be in type 1 diabetes research in the UK. And in the spirit of kind of team science with this, I will present some of the data in a little while that's unpublished from my team, which we think has got significant implications for how we might think about and treat type 1 diabetes. So let's take a step back for those of you not used to looking at the pancreas. And I just want to give a little bit of background, but I will go through this fairly quickly. So if we start looking at a type 1 diabetes timeline, we know that some people have a high genetic risk of developing type 1 diabetes, and that these individuals appear to be exposed to some kind of trigger, which could be something like an intraviral infection that can get into the pancreas and infect the pancreatic beta cells, and that this, together with the genetic risks, leads to the development of autoimmunity. And we can pick that up in the blood. So for example, we can identify islet-specific autoantibodies, and we can look at islet-specific immune cells in the blood that tell us that this process has started in these individuals, but has not necessarily developed towards clinical onset. Once that process has started, we understand that there is a degree of beta cell destruction or dysfunction that's happening, and you have to have sufficient of this before we actually see clinical onset with the symptoms associated with the development of type 1 diabetes. So we know a lot about genetic risk now, and more of that is emerging, and a lot of that work's done by people on the call. We know a little about triggers. We know a lot about autoimmunity, and the team in Bristol and sort of down in Exeter are doing a lot of work on this. But what actually happens in the pancreas in this area is relatively a bit like a black hole. We don't know, a very grey area, and that's mainly because of the availability of human pancreas for research, for us to try and look at that. So there are fewer than 700 type 1 diabetes pancreas reported worldwide, and less than 400, maybe a few more now, are available for research. And that's predominantly because the pancreas is located deep in the abdominal cavity, and doing biopsies is associated with lots of adverse outcomes, and therefore not advised. So sadly, we only get pancreas for research when an individual has died. So as I mentioned, we have relatively few pancreas from individuals with type 1 diabetes, but we also have relatively few from individuals without diabetes. And it is as important to study those without diabetes as it is those with diabetes for us to understand what's going wrong. So we are very lucky in Exeter to have access to this Exeter Archival Diabetes Biobank, which we manage in Exeter. And this biobank consists of a large number of pancreas donor tissues from individuals who sadly died close to onset of type 1 diabetes. But we also access a whole range of different pancreas biobanks through our collaborations around the world. And this probably means that as a team, we've then examined more type 1 diabetes pancreas than anyone in the world, and probably also a large amount of the non-diabetic pancreas as well. So to orientate you and to help you to kind of become familiar with what we're looking at, a typical pancreas section will be collected. So tissue will be collected from the pancreas, and then for the purposes of what we're doing, it's formalin fixed and embedded in these wax blocks, and then we can cut these very thin sections and stain according to the, for the markers that we want to look at. And this is what a typical pancreas section might look like, where we have the acinar tissue that you can see in the kind of blue here, kind of almost looks like a C. This is the part of the pancreas that produces all of your digestive enzymes. And then we have these sort of small islands, which are the islets of Langerhans, and these contain all of the endocrine cells that help to produce your blood sugar, regulate your blood sugar. And I kind of think that these look a little bit like islands in a dangerous sea of these cells that make digestive tissues. And that's really not too far from the truth, actually, when we look a bit closer. So in a healthy islet, it is almost like an island fort in that they have defensive walls around the outside to help protect them from that, the asana cells that are surrounding them. And you can see that clearly here around this particular islet. We also know that these highly healthy islets have a diverse community within them. So we have the 5 different types of hormone producing cells that all live in this community within these islets, and these each help to control blood glucose levels in different ways and communicate with one another to regulate their function. We also know that the healthy islet requires transportation. So we need to get oxygen and fuel in, and we need to get the endocrine hormones that are being produced and the waste products out. So we have this vasculature that comes into the islet, feeding those cells with all the fuel that they need, and then that those hormones and waste are released and taken off elsewhere around the body as required. And finally, they also need charging. So they have a power supply effectively with the innovation that will come into the islets. They enter the islets and can provide signals from the brain that can turn specific cells within the islets on or off, depending on the stimulus that's received. And so, you know, these islets really do look a little bit like forts in a kind of hostile asana sea that we see. It's also important, I think, to highlight that in humans, the islets don't look exactly the same as they do in mice. And many studies are out there now to show that we can cure type 1 diabetes in mouse models, and those haven't always translated brilliantly into humans. And I think that's because there are some fundamental differences in the pancreas structure. So, for example, that peri-islet basement membrane I was telling you about looks very different in the mouse compared to man. I won't go into the details of it, but the constituents of it are different, and there are actually 2 layers in man compared to only 1 layer in the mouse. We also know that the architecture of the islets is subtly different between mouse and men, where you have a core of beta cells with the other endocrine cells around the outside, where it's more interdispersed in the humans, and this could impact on what's happening in there. That vasculature, so that transportation system, is also different, where in mice you have a feeding arteriole that comes into the pole and then it kind of wraps its way around the islet, whereas in humans the feeding arteriole appears to come into the core of the islet and sort of spread out almost like a tree. And finally, the innovation, that power that we come in, again, it tends to track along the vasculature, coming in at the poles of the mouse islets, but into the core of the human islets. So it's important that we recognize that there are differences between man and mouse, and that might explain why some of the studies have not translated so well, and highlights really the importance of trying to understand what's happening in the human pancreas as well. So what do islets look like in type 1 diabetes? So this is the pancreas from a short duration donor with type 1 diabetes, and we've stained for 3 things here. In pink, we have a stain for insulin for the beta cells. Glucagon identifies the alpha cell, so one of those other endocrine cell types, and CD45 is in brown, which stains for the immune cell. So we can see a pancreatic lymph node at the end here of this particular section, and what you can see is it looks very different to that first one I showed you from the individuals that did not have type 1 diabetes, where all the islets or the islands that you're seeing there were kind of both pink and blue. What you can see are a few areas or lobes within the pancreas where the islets look relatively normal, but in other areas we can see only blue staining. So that is areas where the immune system has come in and has actively destroyed all the insulin producing pink cells that we've got there, and have now left. But in this particular donor, and what makes studying short duration donors so important, is we can see around some of these islets, we can see the brown immune cells that are infiltrating around the outside of these islets. And this is where we can actually study the active destruction of those beta cells and learn a little bit more about the underlying process. And so by looking in those pancreas samples and taking into account that in those donors, we can identify different islets at different stages of the disease process, we can start to put a timeline on the destruction of the beta cells and learn a little bit about what's happening at different stages, which are the key immune cells that are present, for example. So we can identify islets where there looks like there's an early scouting party. So this is mild peri-insulitis, and hopefully you can see one or two of those CD45 positive immune cells sniffing around the outside of that islet basement membrane. Those forces start to mass. Again, they are prevented from gaining access to the islet by that islet basement membrane, but eventually those forces become too much and they manage to break through that islet basement membrane, enter the islet and destroy the beta cells. And once the beta cells have all been destroyed, you can see no pink cells left here, there's no longer any signal remaining to bring those immune cells in. So those immune cells then will leave from and go off maybe to identify other islets. So by taking these precious samples, we can learn a lot about the disease process. Even though it's from an individual at one time point, we can at least put some timeline on there. We can characterize the different types of immune cells that are present at the different stages, and this can help give us clues as to which immune cells might be providing the signals, for example, to get through the islet basement membrane, and so on. So I'm not going to go into sort of more of the details of that today, but I'm just going to give you a brief overview of the kind of areas that my team is working in. So we're using these unique pancreas biobanks. We're doing targeted staining, and then we can scan these sections and we can stain either bright field or fluorescence. We can generate image analysis pipelines to ask lots of different questions. For example, that pseudo time analysis that I was talking about, we can look at when we're losing particular types of cells, all sorts of different questions. We can look at the islet basement membrane and as that is, and when that's being broken down, and relate that to which different cells are present, and that sort of work that we're undertaking. So within my fellowship, I am actually feeding into at least two of the grand challenge areas of focus. One is the root cause, looking at whether we may have a period of risky development, and I'm going to tell you more about that in a moment. But we're also looking at rescue and thinking about strategies that might be able to enhance islet defences. And our hope is that we can feed that information into the theme that's focused on generating better stem cell derived beta cells, because if we can give them information on how they can protect those beta cells when they put them into donors, hopefully the outcomes will be better. And then we are using our pancreas biobank as a translational resource by working with lots of teams across the world to try and help them to answer the particular questions that they have that they need to check out in human tissue. So the main focus and the new data I'm going to tell you about today is looking at the heterogeneity of type 1 diabetes. So we all know that you can speak to people who have type 1 diabetes, and some people were diagnosed when they were very young, and they have a very different journey with their diabetes compared to someone who is diagnosed a bit later in life. So the clinical kind of challenges that these individuals face appears to be very different. And in studies that we have done with P-Elite in particular, we have been able to show that there is heterogeneity present in the pancreas of individuals with type 1 diabetes that aligns very, very nicely with that those different ages of onset and the kind of clinical course. So we have put forward this hypothesis that this pancreatic heterogeneity that we're seeing may be related to the maturity of the pancreas at the time that autoimmunity or the first signs of the disease is initiated. And in order to kind of put that into context, we need to think about the fact that the pancreas grows hugely, massively in size over the first 20 years of life, and then it kind of levels off into adulthood. But one of the biggest periods of time where we see the most dramatic changes in the pancreas weight is in the first 2 years of life, where it's a really, huge jump, where the pancreas is changing its whole architecture and growing massively. And that period of time coincides with the times where we start to see the development of islet autoimmunity and some differences in that. So if you are an individual who seroconverts shows signs of islet autoantibodies in the blood at a very young age, your 5-year risk of developing type 1 diabetes is very high. And by definition, then, their clinical onset of disease is likely to be very high risk by the age of 7 years. And this lines up very nicely with the pancreas pathology that I was talking about, where we've defined this thing called type 1 diabetes endotype 1. I'll tell you a little bit more about that in a second. Whereas if you seroconvert a little bit later in life, your 5-year risk of developing type 1 is lower. So you have a moderate risk of developing type 1 diabetes by the age of 13 years. And this seems to associate with our type 1 diabetes endotype 2. So what we're essentially asking is whether early life pancreas growth and restructuring could be impacting the development of type 1 diabetes. So I've mentioned these type 1 diabetes endotypes that Pia had described during her PhD and her postdoc, and is further exploring with an R.D. Lawrence fellowship now. But essentially what I want you to remember is that TIDE 1 is associated with very young onset of type 1 diabetes. There is much more evidence within the pancreas of an aggressive immune response, and at onset there are very few remaining beta cells in these individuals, and they are lost very rapidly, with the majority of them gone by 1 year post-diagnosis. Whereas individuals who appear to have this TIDE 2 tend to be older at onset. We find much less evidence of large numbers of immune cells present within the pancreas, and they have lots of beta cells at onset of disease, and they retain these for a long period of time. So we've got these big differences in what the pancreas pathology in terms of type 1 diabetes looks like, and we're trying to map that back onto the maturity of the pancreas at the initial onset of autoimmunity. The second thing I want you guys to be aware of is this, and if you haven't read it and you're interested in these types of things, this is a great paper that was published by a guy called Ulf Algren beginning of this year. And they performed 3D volumetric analysis of the endocrine mass throughout the entire human pancreas. So this is the first time this was done. There's lots of interesting findings in there, but 2 in particular I want to highlight. So when they sum up all of the endocrine objects, so those little islands that we were looking at, they find that around 40 to 50% of them are very small, so are single cells or clusters. And number 2, they were able to show that those small clusters are predominantly made up of beta cells. So their study was completed in only 5 adult pancreata in 3D. So what we wanted to try and do in this study, and to map it back onto what's happening to pancreas architecture very early in life, is to see if this holds true in 2D analysis, and what happens during aging and disease to this composition of these different endocrine objects. So in order to do that, we have accessed tissue from those different biobanks I've been telling you about, both archival and contemporary. And in a sense, we've gone back to basics and accessed already stained tissue sections that have either had a standard H&E stain, which is a kind of standard histological stain done on many tissue types, or we've stained for chromogranin A, which will pick up our endocrine cells, or sections have been stained to detect either insulin or glucagon to enable us to pick up either the beta cells or the alpha cells. And by going back to basics and using these already stained sections, this has enabled us access to thousands of slides. We can go back into donors that we no longer have any tissue available on and still extract data from them, we can start to look at multiple different regions across the pancreas, because we know from other studies that it's not the endocrine objects are not consistent right the way through the pancreas. We can look across those different ages, as we've been mentioning, so looking at what's happening in very early life and in people with and without diabetes. And we can even now start thinking about if there are any ethnicity differences, which hasn't been explored before. We're then using these AI-assisted pipelines to enable us to interrogate what's happening within the pancreas. And essentially what we do is we teach these pipelines what the architecture of the pancreas looks like, and then we can get a range of different outputs that can tell us about the different endocrine objects, how many there are, what area they make up. We can also use these pipelines with these positively stained immunohistochemical stains to accurately quantify and enumerate those different objects as well. We then had to go through a series of different quality controls to check that these pipelines were performing very well. And we did this in quad donors, where we had serial sections that stained for chromogranin A to pick up the endocrine cells shown in the dark blue here, and the H&E pipeline, where we have the standard histological stain, but we're using the kind of way we've taught the AI how to recognize the architecture to find the different endocrine areas. And we can see that we've got very good correlation between those, and then we can get a whole range of different outputs out that we're starting to interrogate, the most important of which for this talk is endocrine area and the islet size, islet density, and beta cell areas and percentages. We have I just want to familiarize you with our islet bins. And what we've done here is we've transformed the islet area, or the endocrine object area, into a easier readout of size. So essentially this data is transformed, and an islet bin of 0 basically is 1 to 2 cells, whereas an islet bin of 3 is 8 to 16 cells. And essentially the larger the islet bin, the bigger the islet. And it just enables us in a minute, when we start looking at some more graphs, to really visualize what's happening as the islet changes in size. So an example of how that works is shown here. So if we compare chromogranin A, which picks up our endocrine cells, and the H&E pipeline on serial sections, what we see is that the chromogranin pipeline is able to pick up lots of those very small endocrine objects that are in bin sizes 0 to 3. So this is sort of 1 to 2 cells to kind of 8 to 10 cells, for example. Whereas the H&E pipeline is actually not very good at capturing those very small clusters of cells. And that's kind of not surprising, because there's no positive stain in there, and they're actually really hard to spot, even by eye. But it is good at is picking up the larger islets, and it's very accurately maps onto what the chromogranin A pipeline picks up here. So these pipelines are performing very well. What this also shows is that it fits with Ulf Algren's data, suggesting that there are a lot of these sort of single cells and small clusters present within the pancreas, and we'll explore that further in a moment. The other thing I think it's worth highlighting is that most of our functional studies that are done on islet isolations are performed on anything that is in an islet bin of 4 and above, because essentially because of islet isolations, you actually lose all of these sort of single cells and clusters throughout the islet isolation process. And most histological studies also focus on these larger islets that are in islet bins 4 and above. So we have lots of these single cells and clusters that are present within the pancreas, and we haven't looked at them. So this is a real black hole, if you like, in our knowledge, real gap in our knowledge as to what these single cells and clusters do in the pancreas. So I'm going to tell you about 5 of the key results that have come out from the studies that we've completed here. So the first point is that what we see when we look at donors over the first sort of 20 years of life is that there is a dramatic reduction in the endocrine object density shown in this graph, or the endocrine area in the tissue samples. Examples are shown here. So blue is the little endocrine objects in a fetal sample here, and a 9-year-old donor here. So we observe that there is a reduction in both islet density and endocrine area in the first 10 years of life. And this is really coinciding with the asana expanding in size. Number 2, when we look at donors with increasing age and we use the islet bins to look at either the frequency of the different endocrine objects on the left, or the contribution of each of those different endocrine objects to the overall endocrine area on the right, what we observe is that there is a shift towards larger endocrine objects with age. So the fetal samples are shown in purple here, and as you get increasingly higher age, and these are all non-diabetics, they go towards the green. And what you can see is that there is a shift towards these bigger islet bins with increasing age. And it's important to note that a large proportion of the endocrine objects, though, are small. So around 40 to 60% of them are small, but they take up ever smaller proportion of the endocrine area with increasing age. So essentially what we're seeing is that you've got kind of these single cells and clusters, and they are developing as you get older into the larger islets, which kind of makes sense. As you might expect. And we have now replicated that in a second, more contemporary no diabetes cohort, seeing exactly the same things. And the most important thing here is that the most dramatic changes that are occurring in the first 2 years of life, which, as I've shown you, is a key point where autoimmunity can initiate. In Alf's paper, what they did is they also separated their endocrine objects into 3 groups. So either insulin positive only, or just beta cells on their own, which are shown in red here, are typical islets, which are insulin positive and glucagon positive, shown in grey, and then islets that have no insulin and just have glucagon present. And what you can see, and what Alfred reported, is that around 40 to 60% of those total endocrine objects are just beta cells only. But as you might expect, with them being quite small, is that they only take up a relatively small amount of the total endocrine volume that they were assessing. So we've now confirmed that that profile bears out in both pediatric and adult non-diabetic cohorts from both EADB and NPOD in the 2D sections. And we've gone on to further confirm that these really are single cells or small clusters by looking at the insulin positive, glucagon negative events in relation to the islet bin size. So here, those red events, we've transferred those into here to look at them in relation to the bin size, and we can see that there's lots of them and they're very small. Excuse me, and that as you might expect, they take up a relatively small amount of the total islet volume, whereas our typical islets are the ones that make up the majority of the total islet volume, or islet area, sorry, and these are in the bigger islet bins. And as we would expect in an individuals without diabetes, we see very few of these objects that are only positive for alpha cells. So the small clusters are predominantly made up of beta cells, and we've confirmed that in multiple cohorts. Of course, the most interesting thing for us is to find out what happens in type 1 diabetes. And what we see is that the single beta cells and those small clusters of beta cells are dramatically reduced in people with type 1 diabetes. And again, we have been able to confirm that in multiple cohorts. So we can see that red bar that was up here in the non-diabetic controls is dramatically reduced in individuals with type 1 diabetes. If we look at that in relation to the islet bin data now, in terms of the fraction of the total islet area, what we can see is that the small beta cell clusters here are virtually absent in individuals with type 1 diabetes, but the larger insulin positive, glucagon positive islets appear to be protected, as we're actually seeing a very slight shift in the size compared to individuals without diabetes. Interestingly, the insulin deficient islets in these donors, which tend to be older at onset in this particular cohort, are actually still quite large, which is surprising when we consider that these islets have apparently lost all of their beta cells. So this could point towards some dedifferentiation and transdifferentiation pathways in these donors. So up to this point, we've been able to show you that small beta cell clusters are preferentially targeted in type 1 diabetes. And I've already shown you that the young donors without diabetes have a higher proportion of these smaller clusters, which we think are in the process of developing into larger islets. So if islet autoimmunity is targeting these single cells and clusters and it initiates earlier in life, we'd hypothesize that there are fewer larger islets in individuals with type 1 diabetes. So to look at this, we asked if the larger islet density is reduced in individuals with type 1 diabetes by specifically looking at the islet density in those larger bins. And that's exactly what we can see in multiple cohorts assessed in different ways. We see a reduction in the larger islet density shown in the yellow here, in individuals with type 1 diabetes, particularly those with the younger onset and short disease duration. So this really supports the conclusion that the loss of the single cells and small clusters is causing a dramatic reduction in total islet density in type 1 diabetes. So I'm going to try and put that together just to show it in a slightly different way. So what we think we're seeing is that in young children, we're seeing lots of single beta cells and small clusters and these small islets present, and they're all kind of packed in quite close together because the pancreas has not yet expanded. And there are much fewer of these larger islets that have this intact basement membrane that I was telling you about at the beginning. Whereas in an older child, we're still seeing lots of single beta cells and small clusters and these small islets, but they've all kind of moved apart a little bit further. They're less dense, and we are seeing more of these larger islets that have got an intact basement membrane, which we think can help to protect them against immune attack. When we map that onto what's happening and looking in our type 1 diabetes donors, what we're seeing is that the single cells are gone, and all small clusters of cells that have lost all their beta cells, and we've just got a few of these alpha cells sitting in small clusters. We observe immune infiltration in the asana part of the pancreas, and in the few islets that are remaining, the density of those is reduced. We can see evidence of immune attack on those ones that still have some residual beta cells. And because these single cells and small islets have been picked off by the immune system, they don't have the opportunity to grow into those larger islets. So we see a reduced density of the larger islets in these individuals. Whereas individuals who develop diabetes a little bit older, we still see the complete loss of the single beta cells, and the small clusters only have the alpha cells. And we do see a reduction in the density of the islets, but there are more islets because they've had an opportunity to make more islets before the immune attack started. And they've been able to generate that islet basement membrane around the outside. We think they're able to kind of fend off that attack from the immune system, and so they can retain those beta cells for longer. And we think this maps very nicely onto the pancreatic endotypes and has got some important implications for treatment. So honing in on those endotypes and thinking about duration as well, if we split that data down a little bit further, and in these graphs, we've got individuals at different ages shown in the grey boxes up here. Those without diabetes are shown in purple. Those who have who are diagnosed under the age of 13, so more likely to be our Type 1 endotypes with either short disease duration or long disease duration, are shown in the sort of darker blue and green, whereas those diagnosed later in life with either short or longer disease duration are green and yellow. And what you can see is that there is very little insulin positive area in these individuals who are diagnosed very young. And then when we start to see those individuals with long duration here, you can see that they've lost all of their beta cells. But as we start getting into age groups where we've got that older onset, we can see that those beta cells are preserved, and we have this shift towards the bigger islets looking like they are protected. It is reduced compared to the controls, but we are seeing a preservation. So it does fit very nicely with our endotype work, and really sort of hints that the absence of these small clusters and insulin containing islets is far more dramatic in the very young type 1 diabetes donors. So we think this has a range of different implications. The first of which is that this suggests that we might need to consider very early targeted immunotherapy intervention, maybe even at the appearance of autoantibodies, so in stage 1 individuals who are at high risk. The reason being, we want to try and prevent the loss of those single beta cells and clusters, giving that individual an opportunity to develop better, larger protected islets so that they can resist the development of type 1. We think this also has important implications for islet replacement strategies, because in individuals with type 1 diabetes, especially those diagnosed young, it looks like they have fewer islets. So we can't protect or recover what is not there. So these islet replacement strategies may be very important for these individuals because they haven't made enough of their own in the first place for us to protect. We also think this has got implications for our understanding of pancreas growth and the expansion of the asana. We know next to nothing about these, as I mentioned, about these single cells and these small clusters of beta cells. We don't even know if they function the same as beta cells that reside within a typical larger islet, and that's sort of something that we're starting to explore now. So we think this is a really kind of interesting implications, and it's something that the field needs to consider. I want to highlight this is the work of the Grand Challenge team, so particularly Katie and Ty, who have done a lot of this hard work setting up all these pipelines and doing all the QC. I'd like to thank the rest of the PIs within the IBEX team, our funders, and of course the biobanks for supplying this material. And just really to highlight that this Grand Challenge has brought together many different teams from around the UK who are working, who are coming together under the UK Type 1 Diabetes Research Consortium, but are being increasingly funded by this Grand Challenge scheme to work in collaboration with one another, each utilising our different strengths and experiences and sharing information between one another, which is fabulous. So I'd like to thank you all for your attention. Happy to take any questions. Want to acknowledge the donors and their families, without whom none of this work would be possible. And of course, the rest of the islet biology team, who are fabulous colleagues, other colleagues within Exeter, and many different collaborators from around the world. And I will invite any questions.
That was really clear and just beautifully put together, some beautiful slides and illustrations in there. Thank you. Alison, go ahead.
That was lovely, Sarah. It's really nice to hear about your work after so long time. One of the people that's not heard of this story before, so you can rest assured it was new to me. I think it's really fascinating what you've got, you and your team. I'm going to just put on a little bit of a thymic person head on for a second now, okay?
Yep.
So let's imagine we're always aging, we're always changing, and there's always death and regeneration of our tissues. And at least in the mouse, we sort of mapped an early event is the pancreas remodeling at weaning stage, where you get a lot of beta cell death, more than the body can probably cope with, and it can maybe trigger type 1 diabetes, at least the priming event. And I'm really curious about these little single cells an early islets you've got. Is it possible, are you able to look at to see whether or not those, it's going to be very difficult, I'm sure, those little islets, as we call them, those come nice little islets, actually develop into the bigger islets? Okay.
Yeah.
So like a developmental way. So in a young child is the accelerated development of the pancreas that is being shoved very quickly into these bigger islets. Now, if you go down the route saying there's a length of duration of life of any islet in a body, just like there's a length of duration of any other cell in the body, it needs to be replaced. If you're not replacing those small islets into the bigger islets, not because they've been attacked, because something's going wrong with the development, then those bigger islets, that's all that's left. And they are being picked off by the immune system. So some of the big islets that may have no infiltration might just have formed from the little islets coming together very quickly, and then they get picked off. So how are you going to tell the difference between the little islets being picked off by the immune system versus little islets that became big islets and then got picked off?
Yeah, as you can imagine, that is a very, very challenging question to try and address in humans. I think our evidence, when we look at the shift in the proportions of the different islet bins with increasing age, does support the idea that the smaller clusters are developing into the larger islets. But we can't prove that in humans. It's very challenging, and particularly challenging these fixed tissue samples. I do wonder though whether, because they're even in adults, and we're seeing it, and it agrees with Ulf Algren's work, we are seeing a lot of the single cells and the small clusters present in individuals without diabetes in adulthood. So there is a lot of them there. And I guess the question is, are they doing something completely different functionally within the pancreas, or are they a pipeline to make new islets that's ongoing, that has been not appreciated up until this point? And I guess importantly, even in people who develop type 1 diabetes in adulthood, those single cells and clusters have gone. So if that is a supply chain for new islets, if you like, you've taken that out in individuals who get their diabetes in adulthood. So they have only got then what they have made at that point. And if there is turnover of those islets, it could, or even just stress on those remaining islets that could impact on the function of the beta cells, could drive transdifferentiation, dedifferentiation, those types of things. It's a very hard question to answer, Alison. We can only present the findings that we've got and try to interpret them based on that and the observations that we've got. But because of the scale we're looking at, I think we can be reasonably confident that some of these things might be true.
Thanks, Sarah. Colin, you've got your hand up. Did you have a question?
Yeah, no, beautiful story. Thank you very much, Sarah. There's a bit of anthropomorphization going on here, that the little ones are more vulnerable than the big ones, and they've got some kind of palisade around them called the basement membrane. But it's the job of T cells to get everywhere. And that's what they do. They infiltrate, they recirculate. So I guess I'm pushing a little bit to try and think what it is that is different about it. And it's always been intriguing, as I understand it, that I think you and others have said before, that islets get picked off one at a time rather than all of them go together. So it seems very important to try and understand what these signals may or may not be that might be different. Because once the immune system is primed, it's primed, right? And it can then find beta cell antigens anywhere in the body. We've been interested in that, in injecting GAD intradermally and getting T cells to recirculate into the skin. So it's not as if there's, they can run, but they can't hide. So it must be interesting to see what you think that kind of mechanism might be.
So I probably put forward a couple of things there. One is, I wonder if our islets, once they get to a certain size and composition, are almost immune privileged, because we really do not see very many infiltrated islets, or i.e. T cells inside that islet basement membrane. They are super rare to find in human pancreas tissue samples, much easier to find in NOD, but NOD, for example, their basement membrane is very different. We have a double basement membrane in human islets that could work to be a more effective wall to keep those T cells out. So I genuinely don't think that there is an ease of access and T cells can just get in and out of these islets. I think to a degree they may be immune privileged. I think in addition, there is a bit of a battleground going on, because we know that endocrine cells within individuals with type 1 diabetes can kind of raise the white flag. And by that, I mean HLA class 1 hyperexpression, which is essentially the way that you can communicate with a CD8 positive T cell. They do have hyperexpression of that, which would make them more visible to those CD8 T cells. And we can see islets with very dramatic hyperexpression, but the T cells are still sitting on the outside around that islet basement membrane. So they may have the white flag up, but they are still not getting in. But the islets also, I think, are fighting back. So we do find evidence of more negative immunomodulatory signals being upregulated in those residual insulin-containing islets, such as things like non-classical HLA molecules, which are essentially a signal to turn off immune cells, and PD-L1, for example, are upregulated in the islets, in some of those residual insulin-containing islets in those individuals. So I think there genuinely is a battle going on. You've got a wall. I don't think they can get through willy-nilly. I generally think that there may also be some actions by the beta cells themselves trying to fend off the attack and try and prevent themselves from being killed through upregulation of other immunomodulatory agents.
Great. It's a quick comment. You'd have thought there has to be something on the basement membrane. T cells are good at getting through basement membranes. That's what they do. They go through endothelia, etc. There must be something that's a negative signal there rather than within the cells. I know we've discussed before, but maybe the reason you don't find them in the islets is they work very quickly when they get inside. So there's not much time. Once they've been in, the beta cells are dead very rapidly and they're out again. But I just wanted to return to the components on the base membrane. There should be something of interest there.
Yeah, and that is something we're starting to explore, and various groups are trying to look at the islet basement membrane components. And you're right, maybe there's something on there that is a kind of keep it out signal for the T cells. But I would agree that I think once they get in, they do destroy very quickly, because the infiltrated islets are very rare. But so for example, in adults who are diagnosed with type 1, we can see, if you look at all the islets that still contain beta cells, even at onset, only around 20% of them have evidence that they've got T cells around or near them. Other islets next door to it can be hyperexpressing class 1, have got no evidence of immune infiltration. So there could be something to do with the recruitment, and that could be, influencing the amount of T cells that are there. But it does definitely seem like you need to have a critical mass of both T cells and potentially also B cells, because that islet basement membrane can get broken down in the younger individuals, or either it wasn't made in the first place. We don't know that. And we can see a lot more infiltration in those individuals, and a hugely, many more immune cells are in and around those islets in those younger individuals. But yeah, I really don't think we understand quite what's going on there, but I do think there may be some degree of immune privilege, be that through telling signals to tell them to go away, a very effective wall that prevents them from going through. I just don't think we understand enough yet.
Alison.
I'm just following on from Colin said, and we've just said, Sarah, I see a slightly different way possible. Would you think about lymph node? Sorry, my computer's just being nasty. Go away. My computer's being nasty. It always throws out questions, but get your hand down, you're talking now. You do think about lymph nodes and the T cells that go in. Obviously, they have to be very motile, have to go searching through the lymph nodes. Don't just go to one part and stay there. They actually move all the way through the lymph nodes, searching for that interaction with antigen presenting cell. So I see in a way that within the islet too, if you have T cells coming to that islet, I get what Colin is saying. They should get through the double membrane. But if they're not managing to move, there's something about the islet means that they can't move, then they stay and then they go. So you would actually have the impression of an empty, non-infiltrated islet where you don't know, maybe, possibly, the T cells did actually go into that islet and then they simply left because whatever signals are within the islet to allow them to be motile and look for it, their antigen, is it there? So there's something really unique about the islets that are being targeted, that they do have those signals. They do give the T cells time to be motile, bind the antigens and start destroying the beta cells. I mean, it's amazing what you do. And it's really difficult what you do because you're just working with these sections. And we keep putting these horrible mouse people, put these really complex questions to you. And you think it's not really fair, is it? But I think it's fascinating. There's all these possibilities of what could be going on. And the challenge for us all is, how do we develop tools to address that? Whether it's using transplantation processes to humanize mice, I really don't know. Would figure it out.
I think there's a lot of challenges there. And I think, I think if your kind of in-out hypothesis was true, I would expect to see more T cells, at least, even if it's small numbers, I would expect to have seen more T cells in some of those residual insulin-containing islets. And in answer to the kind of going in and out, I remember having a conversation with Alan Fowlis, who's pathologist, who gifted these samples to us. And he was of the impression that what happens is that T cells are coming in through the vasculature and they're going through the islets and they're probably getting the signals that something is not right here and that they need to extravasate. But perhaps because of the islet basement membrane or some other kind of component that we haven't yet identified, they cannot extravasate until they are actually outside of the islet periphery, which is why we then see them coming back around and sort of sitting at that islet basement membrane around the outside of it, kind of trying to get in. We also know that there is a lot more inflammation in the young children in the asana. So if you have got those single cells and clusters sort of dotted throughout the asana tissue, those T cells that have extravasated, they will be able to be motile and moving around in the asana, picking off those single cells and clusters. So I think they are going to be more susceptible to that destruction, and that the bigger islets, and we're certainly seeing a shift towards the bigger islets are the ones that we keep for longer. So it does suggest that something about those bigger islets and some constituent of those bigger islets is able to kind of, but fend the T cells off for a bit.
Thanks, Sarah, and that's been really fascinating, and I hadn't even noticed it's gone 4 o'clock, so I better let you all go. No, thank you. It's been really great to hear about your work, and I'm sure everybody's really enjoyed it. So we'll hope to see you all again on the 18th of November, if not before.
Everyone.